Okay, thank you, Dagnia. It's a great pleasure to take the opportunity today to spend some time with you, especially on a beautiful afternoon like this. I want to thank you all for coming to the laboratory. It's really a great pleasure for me to work here at Cold Spring Harbor Laboratory and to be part of the very close relationship that the laboratory has with the community, which is really unique. When we have visiting scientists come here from around the world, one of the first things they tell us when we talk to them is that they admire us for this relationship that we have. And this lecture today is part of our agenda, as Dagnia pointed out, to continue this relationship and discussion with our neighbors so that we can together help move science ahead and cure disease. Now, today, what we are going to focus on, if you can turn the lights down, is this amazing book that Rebecca Skloot wrote and put out in February of 2010. This book is soon to be out for 3 years, and I just checked this morning, and the paperback copy is still on the New York Times bestseller list. So it's been on the New York Times bestseller list in one way or another for practically 3 years, which is quite remarkable. And so the first question we should ask is, why is this book so popular? Usually books about science are only popular to a very limited audience, yet this book has had widespread interest among people of various walks of life. And there are really 4 reasons that I could think of that makes this book of interest to so many people. One is because it's about race. 2, it's about ethics. 3, it's about science, and 4, it's about doing the right thing. And what I'd like to do today is to touch on all 4 of those aspects of the book. So to start out, there are 2 characters in the story. One, of course, is Henrietta Lacks, and the second is the HeLa cell that was derived from a tumor that she had. So I thought to start out, we should talk about a cell. And this is a cartoon schematic of a cell. And as you probably remember, almost all cells in your body have a nucleus. And the nucleus is where the DNA is. And the DNA is actually the instruction book that allows the cell to produce RNA molecules, which in many cases will be translated into proteins, and in some cases will work specifically as RNA molecules. But together, all of these components will allow the cell to function. There's only one cell type in your body that does not have a nucleus, and that's the mature red blood cell that's circulating around in your arteries and veins. And the reason is that the mature red blood cell has one function, and that is to deliver oxygen to all of the organs in your body and to remove carbon dioxide and bring that back to the lungs so that it can be expelled. Now, in the cytoplasm of every cell are a series of different organelles, and these organelles are taking part in a variety of processes, including making proteins, respiration of the cell, etc. And together we have this little machine that communicates to other cells and also works on its own and allows us to survive as we do. Now, cells come together and make a structure that we refer to as tissue, and tissues come together and make organs. The average human has about 60 trillion cells in our body, which is quite remarkable, comprising about 210 different cell types and 78 organs. Normal human cells have 46 chromosomes. However, HeLa cells have about 82 chromosomes. So that immediately points out some major problem, because the chromosomes contain genes, and genes encode for proteins. And all of this process has to be regulated in the cell. And if a normal cell has 46 chromosomes and works properly, and a HeLa cell has 82 chromosomes, you can already begin to imagine that things are going awry in this HeLa cell. It's not doing a lot of things the way a normal cell does. It may be accentuating certain processes. It may have eliminated certain processes, but it is just a cell that really is going off in all angles and creating a lot of problems, and we're going to talk about those. Normal cells were first grown outside the human body in 1907, but they died after they divided about 50 times. And this was a big, big problem for scientists, because if we're going to understand how cells work and how humans work and why we get disease, we need to have a model to study this. And the most basic model is the cell. And so we need to have cells that we can grow in culture in many, many divisions, much more than 50 divisions, because we have to do lots of different experiments and we have to follow things over time. And we can't be limited by just 50 divisions if we're going to understand how cells and how organisms work. HeLa cells, however, have been growing in culture for over 60 years. And so you can see that they have provided us with an amazing system from which to study basic cellular processes. While this whole idea of getting a cancer cell to grow in culture was a major undertaking in 1951 when these cells were isolated from Henrietta lacks, currently we have over 2,000 human cell lines available that can be grown in culture. So we've overcome a lot of obstacles over this time period. Now, we should talk a little bit about the scientific landscape in 1951. And as you know, context is extremely important. No matter what we talk about in life, the contextual setting of the process, whether it be scientific or not, really impacts significantly our understanding and our interpretation of the events that occurred. Now, in 1951, as I already mentioned, cells could not readily be grown in culture for long periods of time, yet we needed this to happen. The double helix structure of DNA that was identified by Jim Watson and Francis Crick was not yet revealed. It was not until 2 years later, in 1953, that this structure was actually revealed in a talk that Jim gave here at Cold Spring Harbor. Cancer-causing genes, which are known as oncogenes, had not yet been discovered. So we knew very little about cancer. We were thinking of it in such a primitive state that looking back from today to the way scientists were thinking about cancer back then is just totally amazing for me. It was just so basic. Hard to believe that they could have even imagined that they might make any major advance in terms of curing this disease or treating it. The first human gene was not yet sequenced. That did not happen until 1972. Human gene cloning had not been carried out. That didn't happen until 1978, and I should mention that many of the major milestones in gene cloning took place right here at Cold Spring Harbor Lab. Human oncogenes had not yet been cloned. It wasn't until 1982 that Michael Wigler here at Cold Spring Harbor Lab and several other investigators, including Bob Weinberg up at the Whitehead Institute in Boston, identified the first oncogene. And again, I can remember to this day when that paper came out. This was one of the biggest advances. We thought that this was the gene that was going to cure this devastating disease. Back then, we thought of cancer as one disease. As you know, today, that is no longer true. Cancer is many different diseases, and even a particular type of cancer is comprised of many different diseases. And finally, to really put it in perspective, gasoline was 19 cents a gallon. So I think that really sends the message home to you of how different the times were in 1951, not only from a scientific landscape, but also for our everyday lives. Okay, so now let's move on to talk about Henrietta Lacks and the HeLa cell phenomenon. Henrietta was a 30-year-old African-American woman, and as it turns out, who lived in Baltimore, as it turns out, the Johns Hopkins Hospital at the time was one of a few that would treat African Americans. Quite remarkable, thinking about that now. In February of '51, Henrietta presented at the Johns Hopkins Hospital with what turned out to be a very large cervical cancer. By October of that same year, she died. And so again, this brings home the idea of how severe this cancer was. And because it was so severe, that's why these cells were able to get put into culture so easily. Okay? I think of a HeLa cell as a weed, right? You have weeds growing in your garden. You don't have to put fertilizer down for them. You don't have to treat them in any special way. They can just grow anywhere. And that's how I think of a HeLa cell. While it does need nutrients to grow, it didn't have a lot of the requirements that your typical normal cell had and that other cancer cells had. And so this put it in a big advantage when scientists were trying to get it to grow in culture. It was a big plus that hadn't been seen before, and it was due to the severity of this particular cancer. Her cancer was actually identified by a very famous physician, Dr. Howard Jones, who was a renowned gynecologist and who later actually performed the first successful in vitro fertilization in the United States, and this is his claim to fame, actually. Now, George Guy was a cancer researcher at the Johns Hopkins University, and it was a routine phenomenon back then for George to obtain cells from surgeons performing operations to remove tumors from patients. Okay, so it was just the common practice. The tumor came out, some cells were taken out of it, they were passed on to George Guy. He was focused on trying to culture cells, and he would put them into culture and try to add different nutrients to the culture medium to help stimulate them. They would generally grow for a while, and then they would die. Why was he doing this? Well, because, again, context is important. Back then, the thought was that if we're going to understand cancer, why does it form, how can we treat it, how can we possibly prevent it, we'll have to get this knowledge from the cancer cell itself. And the way that we can do this is to get this cell to grow in culture, and then we can manipulate it, we can see how it grows, we can understand the critical factors that will allow it to grow, that will inhibit it from growing, etc. So this was a really, really big focus of cancer research back in the early 1950s. Now, as you know, George Guy successfully put these cells into culture, and he was actually astonished by this because he had been trying for so long unsuccessfully. Once he got these cells in culture, he was so thrilled about this that he immediately began to share these cells with scientists around the world and sent out cultures of the cells from Johns Hopkins University for no fee to any scientists who requested them in the effort that he could help to advance our understanding of cancer and work toward a cure. So now I thought I would show you, who are these cells? What do they look like? So this is a beautiful micrograph taken from a good friend of mine, Tom Durink at the University of California, San Diego. And what Tom did was he grew some of these HeLa cells in culture, and he stained them in 3 different colors. Okay, cells usually don't have color, okay? They're colorless. But he was able to label different structures in these HeLa cells with different color dyes. And so these blue round structures or oval structures are the nuclei of each of the cells, and this is where the DNA is. The green structures out here in the cytoplasm are microtubules. They're filaments. You can think of them as architectural elements that help give shape to the cell. And finally, the third structure is actin, which is another type of filamentous structure. And you can see the actin filaments out here, again, in the cytoplasm. And it's these actin filaments that help the cell to move around and also act as a framework for other molecules in the cytoplasm to move on. This is another image of HeLa cells. Again, here you can see two different HeLa cells that are in what we call interphase of the cell cycle. That is, they're not dividing, okay? But cells divide usually once every 24 to 36 hours. And when cells divide, the genes on the chromosomes in the cell condense. And here you can see the condensed chromosomes in this cell. The membrane that surrounds the nucleus breaks down, and so now there is no separation between the nuclear contents and the cytoplasmic contents, and so you can see the chromosomes. Now, it's very easy to talk about cell division and how it happens, but perhaps it's a bit hard to imagine how it happens. And so I wanted you to really see how cells divide. And so I asked two scientists in my lab, Ingling Kumaran and Stephen Hearn here at Cold Spring Harbor, if they would film HeLa cells so that you had the ability to actually visualize them over a period of about 87 hours, okay? And so this is going to be a movie that we're going to watch. And what they did was they took a Petri dish and they added some media, which contains nutrients, to the dish, and then they added some HeLa cells to that dish. And you can see individual cells here, but you can see that a large portion of the dish has nothing on it. And we wanted to have a lot of empty space to give these few cells the room to divide and then to fill up the dish. And so when I start this movie, we're going to begin to watch cells as they go through the cell cycle. And I'll point out a few interesting phenomena. So you can see that the cells, first of all, are not static. They move around, and you will begin to see a cell round up and then divide, divided into two. And if you follow them, you'll continuously see that they divide. You'll also notice that occasionally a cell will die, like right there. They go into a process called apoptosis, cell death. And that might be because they got the wrong complement of chromosomes. It might be because they don't have enough media or nutrients, etc. So you continuously see them dividing here, and you see that they're filling up the dish as they divide. They're also moving around on the dish, and we're going to watch a cell up in this area in a few minutes, and it's going to divide into 3 instead of 2. It's going to be somewhere around here. It'll be a rather large cell. There it goes, right here. There, see, it went into 3. This is really bad news, okay? Because when a cell divides, it has to give equal numbers of chromosomes to each daughter. If you have a third cell, that means somebody's not getting the right material, right? One cell may be getting more chromosomes, one may be getting less, etc. So now you're immediately setting yourself up for problems. These cells are going to have things going wrong with them. And this is what happens many times with these cancer cells. They don't go through normal cell division. They go through aberrant divisions, and that's why I mentioned to you early on that most HeLa cells have about 82 chromosomes. And if you counted the number of chromosomes in a population of 100 HeLa cells, every one would probably have a different number of chromosomes. And so they're really very aberrant cells. Okay, so now that you see how cells divided, let's talk a little bit about some of the major advances of these cells. Now, in some ways, this is sort of a contradictory thought, right? I've just been telling you about how bad these cells are. They're not normal. They're dividing aberrantly. And now I'm telling you that I'm going to talk about the advances that came from them. Well, the reason is that even though these cells have a lot of problems, and in fact those problems allow them to be grown in culture, we can learn a lot from them, okay? And the reason that we can learn a lot from them is they still have to undergo all of the fundamental processes of life, okay? That genes have to get turned on, they have to make RNA, RNA has to make protein, protein has to be transported, communication has to take place between cells. So we can learn an enormous amount from these aberrant cells. And I think it wouldn't be wrong to say that we have probably learned significant amounts of information about every single major biological process from HeLa cells. So quite remarkable, yet quite unique. So early on, HeLa cells were used to actually propagate the polio virus, which then led to the development of the vaccine against polio by Jonas Salk. Okay, so again, the virus was able to grow in these cells and then to break the cells open and release more virus particles. The virus needed the cell as a substrate to allow the virus to grow. The cells were sent up in the first space missions to see what would happen to human cells in space, right? Before we send humans up in space, better check on the cells and make sure that the cells don't explode or something awful happens to them rather than send up a human being. The effects of radiation on human cells were tested. They were used in studies to help map human genes. They were used to prove that human papillomavirus causes cancer. These HeLa cells, in fact, contain human papillomavirus. They were used to help identify the mechanism of tuberculosis and HIV infection. Again, the cells were infected with these viruses. One had this cell system in culture, and one could easily study how does the virus get into the cell. It didn't matter that it was a HeLa cell. It just mattered that it was a human cell, and we need to understand how the virus gets into a human cell. They were used to discover that human cancer cells contain telomerase. This is an enzyme that regenerates chromosome ends and prevents them from aging and dying like normal cells. This is a very important process, and in fact, one of our former faculty members here at Cold Spring Harbor, Carol Greider, won a Nobel Prize for work that she had done when she was doing her PhD in California at UCSF with Liz Blackburn. And ironically, the fact that Dr. Chester Southam at the Sloan Kettering Institute in New York City injected HeLa cells into uninformed human test subjects to see how cancer initiates led to an NIH investigation, that's National Institutes of Health, and the initiation of medical review boards and informed consent by patients. Now this is really a remarkable point. Chester Southam turns out to have been the chief of virology at the Sloan Kettering Institute. And he was a physician who was overseeing patients with leukemia as well as other cancers. And when these HeLa cells came out, everyone was very anxious to try to understand how they caused cancer and so on. And so what Chester did was he actually took HeLa cells and he injected them under the skin of these patients who had leukemia and was able to observe that tumor nodules grew under their skin. Now, of course, this was totally without their consent, but you have to also remember that there were no guidelines in place back then. He was doing these experiments in 1953, 1954. There were no guidelines, so you can decide yourself if this was an ethical thing to do or not. Now, once Chester did these experiments and found that these cells were causing tumors in these cancer patients, he wanted to take it to the next step, and he wanted to see what would happen if he was to inject these cells into the skin of normal patients. So he went and contacted a penitentiary in Ohio, and prisoners in this penitentiary felt that they were doing a service to society by allowing him to inject these tumor cells under their skin. And he found that the tumors grew, but much more slowly, and much fewer tumors grew. Okay? He then left the Sloan Kettering Institute and he went to a hospital in Brooklyn, New York to continue his studies as well as his clinical affiliations. And at this hospital in Brooklyn, he tried to take two of the younger physicians there who were under him into his study that he was doing, and he tried to convince them to continue these injections so that they could study the effect of these HeLa cells in humans. And these two young physicians refused to do these experiments. And he put pressure on them, and they still refused. And finally, it got to the point where these two doctors basically said, "We're out of here. We quit." And when they quit, they sent their letter of resignation to a local newspaper in New York City, and this was published. And I don't have to tell you what happened after that. Obviously, Chester Southam was in a lot of trouble, and in fact, he lost his medical license, but unfortunately, it was only for 1 year that he lost his license. But it was through these awful events that the National Institutes of Health actually realized that there better be some guidelines in place not to allow this kind of thing to happen. You can't basically use humans as test subjects without their direct permission. And so this really represented, as I point out here, this was the starting point, actually, of guidelines for the ethical use of human subjects. And so it's an important point, actually, even though it's an awful point to discuss. That was about 1953, 1954. So it wasn't too long after HeLa cells were first identified. Okay, so now I just wanted to say a little bit about HeLa cells in Cold Spring Harbor Lab. I told you about some of the major events that in science that they played a part in. I want to now tell you some major events here at the lab that were very positive events, in fact. In 1970, Joe Sambrook, who was at Cold Spring Harbor Lab, actually isolated a native form of a very important enzyme in cells that allows genes to get turned on. And this was very important. And the reason that Joe was able to isolate this enzyme was that he could grow up enough HeLa cells so that he can get the molecules of the enzyme out of those cells and characterize it. And this was a very important early event toward our understanding of transcription. In 1979, Osborne and Weber identified a series of proteins in cells that form filaments. They're not the microtubules or the actin that I showed you. They're a different family of proteins. They're referred to as keratins. And these proteins were later shown to be very useful in cancer diagnosis and staging cancers. So again, a very major advance being made toward understanding cancer from using these HeLa cells and characterizing specific protein molecules that these cells were making. In the 1980s, David Beach's lab at Cold Spring Harbor identified a series of critical proteins that control the cell cycle. Remember, I showed you how these HeLa cells are dividing, and they're dividing every 24 hours. Now, an important understanding of cancer, as tumors grow and become bigger, you want to understand the proteins that regulate that division process in that cell cycle. And David's lab, which was a lab using yeast initially to study these proteins, then moved into the HeLa cell realm to identify the human proteins that were involved in this process and made some very, very important advances. In 1981, Walter Keller's group, again, taking after Joe Sambrook's work, looked at making transcription extracts. That is, transcription is the process of converting a DNA, making a copy from DNA into RNA. He made extracts out of the cells that allowed this process to take place in a test tube. Okay, it allowed people to then study this process in a test tube rather than in a cell, which then allows you to be able to characterize it in excruciating detail so that you can really understand it and then figure out how it goes awry in disease. And then finally, in 1990, Adrian Craner, who is still here at Cold Spring Harbor, purified a factor, a protein molecule, that is critical for the editing of RNA molecules. So when an RNA molecule gets made, it has regions that are really important. They're called coding regions. And then there are regions that link the coding regions that are called introns. And in order for that RNA to make a protein, you have to cut out those introns. So you're editing it, and then you're putting the two coding regions together, and then you make a chain of the coding regions. Adrian identified a very important protein that functions in that process, okay? And he subsequently demonstrated that not only was this protein important for this basic cellular process, but the protein turned out to be an oncoprotein. If you overexpress this protein in certain cells, it will cause cancer. This is a major finding, okay? And the only way that Adrian made this finding was because he was a basic scientist. He was interested in understanding the basic process of the cell, how RNA gets edited. And because he had this understanding and the depth of this understanding, he could then use that and see how that protein might function in the cancer process. So I use this point just to stress how important basic research is. You know, sometimes we think about, well, you know, shouldn't we all be doing research directly focused on a disease with a specific question, you know, honing into that disease? Well, sure, we should be doing that type of research, and we are. But we also have to do basic research, because many times you don't know where that diamond is going to come from. You learn enough about a basic process, it's going to connect the disease. There's no question about it. And this is a beautiful example that happened in Adrian's lab. Now, I want to take that one step further to a really remarkable finding, because Adrian, by understanding this process of RNA editing, has recently used this knowledge to figure out the basis of a childhood disorder called spinal muscular atrophy. And this is an awful disease in kids, and they die at a very young age. It's a muscle disorder. And because of his knowledge in this splicing process, Adrian came up with an ingenious approach where he could alter the editing process in the cells in an animal, okay? And so he made an animal model of this disease, and it had all of the features of the human disease. And then he developed a drug, and he treated the mouse, and the mouse got back its normal ability to move around and walk and use its muscles. It was amazing. Every time I heard him give a talk on this, I was more and more amazed. Well, guess what? This work has, this year, gone into clinical trials at a number of hospitals around the country, one being Columbia University Medical Center in the city. So again, I can't stress enough the fact that this went from basic science to really a clinical situation, and we're very hopeful that this drug will work and move ahead in various stages of clinical trials and be able to help these children with this dreadful disease. And many other scientists now in other fields are saying, wow, that's pretty remarkable. Maybe we can use this approach in the disease that we're working on. And so scientists are looking to see if that approach might be applicable to their disease systems. So again, really a beautiful example. Now, why am I talking so much about this? It's because Adrian had to purify this protein, okay? And he decided to purify it from HeLa cells. And the reason is that the protein is present in very few copies per cell. And he needed lots of cells to get enough of the protein to understand how it functions. And it turns out that he had to get trillions of cells in order to extract enough of the protein, okay? And trillions of cells came from thousands of liters of HeLa cells. So this was an enormous undertaking on his part, and it was only because he could grow these cells to such a high density and in such large amounts that he could have purified this protein. Okay, and then many, many studies have been going on here at Cold Spring Harbor from the 70s to today, using HeLa cells to localize various proteins that we all are working on to see where do they sit in the cell and where do they function in the cell. And so we're basically using the HeLa cell as a vehicle to really understand where the molecules that we're interested in studying reside and how they work. Okay, so this then brings us into this whole issue of informed consent and patients' rights. And we already sort of touched on it a little bit when I talked about Chester Southam and him injecting HeLa cells into patients' skin. But also we have to keep in mind the fact that when Henrietta Lacks walked into the Johns Hopkins Medical Center, her tumor was removed and her cells were given to a scientist to try to grow in culture. She had no knowledge of this, okay? Again, there were no rules back then, so no law was broken, nothing wrong in a sense was done because that was the general practice at the time. No, I'm not saying that that is the right thing to do, but that was the general practice back then. Again, context is important. But what then happened was, it wasn't until 1971 when George Guy retired and there was a ceremony to honor him that it came out in that discussion and papers from that HeLa cells actually came from an individual by the name of Henrietta Lacks. Okay? Up until that point, up until 1971, nobody knew who Henrietta Lacks was. Okay? They used the acronym HeLa. H-E is from Henrietta, L-A is from Lacks, but no one knew who it was. And in fact, in some cases, HeLa was referred to as Helen Lane to keep Henrietta Lacks anonymous. Okay? We would never use an acronym like that today because the acronym would have to be so anonymous that you couldn't even guess who the individual might be that these cells were coming from. But you could imagine how the family felt when in 1976 a story came out in Rolling Stone magazine about this whole HeLa cell thing. And the family found out about this and they were outraged, right? Here was their mom who was in a hospital, had an awful tumor, died, her cells were taken, they were never told about it. They thought that lots of money was made from these cells, and there was a lot of feeling about, well, maybe, part of that should be ours. We're not very wealthy. We can't really afford health care. Why are all these people having all these things with cells from our mom? And so you could imagine, how they felt back then thinking about this whole thing. And it was a really, really hard process for them and a long process for them. I mean, after the fact, in speaking to their sons, Rebecca Skloot acknowledged in her book that they said, we would have been happy to have the cells donated. We just wanted to know about it. And so again, that's where my point about doing the right thing comes up, right? I think everyone would be happy to do something to help their fellow man or woman and society, but you would at least like to know that you have the option to do it and that it's happening. And they were sort of kept in the dark back then. And that created a lot of problems, as you might imagine. Okay, so this whole idea about informed consent and patients' rights really can be carried all the way back to World War II, when doctors in Nazi Germany were conducting research on prisoners in concentration camps. And after the war, these doctors were tried at the Nuremberg trials. And it was from the Nuremberg trials that in 1947, a document came out which is referred to as the Nuremberg Code. And this is an international document stating that voluntary consent is mandatory for any clinical research. So this was a very, very important statement, but also it was not a law. It was simply a statement. There was no jurisdiction in essence, over this point. In 1945 to 1966, in fact, the NIH funded 2,000 research projects, and none of them used informed consent. Okay? 1964 was the next major event where the Declaration of Helsinki was put forth. And this was a set of ethical principles regarding human experimentation, and this was the first set of ethical principles that was put forth by the medical community themselves. So this was a very important and major statement. However, once again, it was not legally binding, and in fact has undergone 6 revisions. So it was simply various groups stating this shouldn't happen, but nobody was overseeing it and making sure that it doesn't happen. In 1966, due in great part to these studies that Southern performed, the NIH came out with a determination that independent institutional review boards must be present at institutions, and these review boards must evaluate any investigator's proposal to do research which is using human subjects. So this was very important. In 1979, the National Commission for the Protection of Human Subjects, known as the Belmont Report, again, another very important report, which was prompted in part by the Tuskegee syphilis study, which again, you might remember was a situation that was horrendous, where individuals were infected with syphilis bacterium, and those individuals that had syphilis were not being treated when we had in hand penicillin, which could be used to treat this dreadful disease. In 1991, the Department of Health and Human Services developed what's known as the Common Rule, and this is a rule overseeing all of this human subjects research, and it was adopted by all agencies that conduct or fund human research. That's above and beyond the National Institutes of Health. And finally, in 2000, the NIH required anyone working with human subjects to take an online course. And I think this finally hit the nail on the head, because now you have to go online, you have to answer all these questions and so on and so forth, and it's quite elaborate, and then you have to put your electronic signature, and so now it is actually documented and is actually policed. And this is taken extremely seriously. We have to have very, very careful and elaborate protocols when dealing with human subjects, human samples, etc. And we are very, very serious here at Cold Spring Harbor Lab, as well as at other institutions around the country for this, because if we fail to do this correctly, we can actually lose our funding. And that would be absolutely horrendous, obviously. So, everyone is extremely diligent. In fact, we have now gone so far that we probably have gone overboard on the other extreme, right? Which happens many times, right? You don't do things right for a while, then there's the big wake-up call, and then you try to compensate, you go way overboard, and then perhaps you have to modulate a little bit, because there are cases now where it might take us a year to get a protocol approved, just because it's going through such scrutiny. And in some cases, it is a bit overboard. So again, just to summarize this, when samples are collected solely for research purposes, an institutional review board must review and approve the research proposal. The wording of the patient consent forms are approved. Every single word is looked at and read and reread and evaluated for every possible meaning and definition, etc. And samples cannot be taken without the patient signing such forms, and the samples are coded to conceal the patient's identity, usually with some random set of numbers which have nothing to do with the patient. It's not their birthday, it's not their social security number, it's not their phone number. Totally random. You would never find out who this patient was. Okay, so that's a whole lot about institutional review boards and human subjects, but there's one more point that we should talk about today, and that is who owns the material, okay? So in other words, the sample is taken, but who actually owns it? And this is a whole separate issue. In 1999, the Rand Corporation actually published a report indicating that more than 307 million tissue samples from more than 178 million people were being stored in the US, right? Whenever you go to a doctor, many times, they'll take a biopsy or a procedure, and then they'll put it into a block, and then they'll take some sections and stain it for something, and then come back and say, oh, you're fine. But then these blocks have to be kept for 10 years in storage in case there has to be follow-up, et cetera. So we're basically drowning in these blocks and samples, and this number obviously is increasing exponentially. So there's a lot of material around. Now, the common rule, which is part of this Declaration of Helsinki, sets forth the composition and function of these institutional review boards. However, this common rule does not address the question of who owns human tissue samples that's used in research, and it does not apply to tissue obtained post-mortem. There is no clearly defined regulation regarding the ownership of human tissue samples and who can control their fate. The ownership question does not depend on whether the patient consented to the use of his or her excised tissue for research. No laws or regulations exist regarding ownership of leftover tissue, fluids, tissue blocks. However, IRB approval is required for their use. Okay? So for example, let's say I'm a scientist here and I want to do some experiments on leukemia cells. Okay, and I contact a clinician at North Shore LIJ, and he says, oh yes, we have these blocks of cells, we can provide them to you. But I would have to put together a protocol that would go through our IRB, stating exactly what I'm going to do with these samples, showing how I will not know anything about the patient's identity. I will have just a random number, et cetera. And so this whole protocol is in place, but certainly I don't need to have the patient's consent to do that. In the case of tissue being excised for clinical purposes or donated for research, courts have determined that patients do not retain ownership rights of excised tissue. So bottom line, once it leaves your body, it's gone. You don't own it. That's the way it is. Although donors have continuing rights regarding the use or secondary use of their samples, they do not own those samples or control their deposition. And so I want to just now point out to you 3 major court cases that have addressed this specific issue or aspects of this specific issue. And the first and most famous is the case of Moore versus the Regents of the University of California. And what this was about was an individual came in to UCLA Medical Center. His name was John Moore. He had a hairy cell leukemia. And as part of his treatment, his doctor suggested that his spleen had to be removed. His spleen was removed, and cells were isolated from his spleen. The surgeon had permission to remove the spleen. Now, cells were taken from the spleen, and they were modified by the physicians and their collaborators to put them into culture so that they could study hairy cell leukemia, okay? But the patient didn't know about it. And so after the surgery and the patient was released from the hospital, he moved to Seattle, and the physician kept bringing him back to UCLA Medical Center for follow-ups. And he kept saying, can't I get a follow-up at a hospital in Seattle? Why do I have to come back to UCLA? And they said, no, you have to come back here. So he was convinced, and he kept coming back, and he kept coming back. Well, after a while, he realized that basically they were just taking more samples from him, and they were monitoring the progression of his disease, both in the laboratory as well as for clinical reasons. So he filed a lawsuit against his physician and UCLA, and it went to court, and he basically lost the case. So then he went back and went to a higher court and tried to get this suit through, and he won the case. And then UCLA went back to yet a higher court, which was the Supreme Court of California. Lawyers made a significant amount of money in this, I would imagine. But in the end, what happened was the Supreme Court stated that any ownership you have in your tissue vanishes when they are removed from your body with or without consent. So that was a very critical case. The second case was a 2003 case, Greenberg versus Miami Children's Research Institute. In this instance, a family came to Miami Children's with two twins who had a very rare childhood disorder called Canavan disease. And it was rare, and nothing was known about this disease. And so obviously the family wanted to help their children. And so in order to help the physician learn about the disease and try to develop a cure or treatment for the disease, the family developed a registry where they could get other families who had children with this disease in, and the physicians could then get samples from those children, in other words, to build up a data bank, because you can't study two people and find a cure to something. So the family helped with that. In addition, the family provided money to the physician to help fund the studies that he was doing, because obviously they wanted to help their children. So they really played a huge role in helping this research. Well, it turns out that the physician actually found what was wrong. He found an enzyme defect in these kids. He filed a patent. The family wasn't informed. And the patent was licensed to a biotech company to be used as a way of diagnosing the disease. So obviously the family was not very happy. I mean, they were happy that a genetic test was now developed, but they were not very happy that they weren't informed about this, and they were not getting any royalties, if you will, from this, since they felt that they provided significant input into this, in addition, obviously, to what the clinicians did. So a lawsuit ensued, and what happened in this case was the court determined that any property rights the patient had were surrendered upon tissue donation, once again, confirming the Moore versus the Regents of the University of California case. However, in this particular case, the plaintiffs received an undisclosed settlement, as the court felt that they played a significant role in getting additional patients to this physician and in funding part of this research. So they got some monetary settlement. Finally, the last case that I want to bring to your attention is a 2007 case, Catalona versus Washington University. And this was a case where a very prominent prostate cancer physician, William Catalona, had a position at Washington University, and at some point in time he decided that he was going to leave that university and go to, I believe it was Northwestern University. So when he moved to this other university, he requested that all of those tissue samples that he had been collecting over the years from these prostate cancer patients be transferred over to this new place that he was at. And WashU said, no way, those samples belong to the university. They were acquired here at this institution. They belong to this institution. You can't have them. So William Catalona then sent letters to every single one of his patients and asked them if they would sign letters saying that they would allow those tissue samples to be transferred to this second university. He got all those letters together, sent them to Washington University, and they said, no, we're not giving them. So this all went, again, to the courts, and finally the court stated, I think I skipped this, that the university, not the physician or patient, owns the tissue samples. So wherever they were collected from is where they belong. Certainly there might be a case where a university might say, okay, fine, we'll give the samples to you, but they don't have to be forced to do it. So this is sort of where we stand right now in terms of tissue. Once it leaves an individual, it's no longer yours. Obviously, it's being used to help society. And so, if you think of it from that good perspective, I think it's a positive thing. So now, finally, I want to end just by saying a few words about Johns Hopkins University and Henrietta Lacks, because as you imagine, as I mentioned early on, there was a very tense relationship when word came out that these cells were taken from her, the family had no knowledge, et cetera, et cetera. And the environment in 1951 was such that race was playing an important issue here. Remember, I mentioned that Johns Hopkins Hospital was one of the few that even accepted African American patients, and so there was a very uncomfortable feeling in many different ways about this whole phenomenon. So actually, I think one of the best aspects of the Rebecca Skloot book, to me, aside from the book being a really fantastic story and everything, is the things that happened at Johns Hopkins University once the book came out, and really quickly once the book came out. So remember, the book came out in February of 2010. Virtually immediately after that, the annual Henrietta Lacks Memorial Lecture was established at Johns Hopkins University to honor her, okay? And her whole family actually was invited and came to that first lecture and have been very, very supportive of this. So they seem to be on board now in lots of these events that are honoring their mom. The Urban Health Institute initiated a Henrietta Lacks Memorial Award in 2011, which recognizes and supports Baltimore community organizations that collaborate with Johns Hopkins University to improve health in the city. Another positive thing, the Lacks Family HeLa Foundation was set up to provide direct help to cancer patients through financial and psychological support. The Virginia General Assembly honored Henrietta Lacks in 2011. The Henrietta Lacks East Baltimore Health Sciences Scholarship was set up just this year, sponsored by Johns Hopkins Health Center to support high school graduates to go to college. And there are educational displays honoring Henrietta Lacks and HeLa cells at Johns Hopkins University Medical Center at an extremely prominent position outside one of their major auditoriums. So again, they've now stepped up and recognized the significant contributions that this individual unknowingly had made through cells that were isolated from a tumor that she had. So all in all, taking context into perspective, of course we wouldn't do that today, but that was the time, that's how things were done back then. I'm not saying it was right, but I think we have to look for the good in all things. And I think in the end, a lot of good things have come from these HeLa cells that have helped move science ahead, that have helped move us toward a better understanding of cancer and even other diseases, because they've allowed us to understand basic processes in cells. And by understanding this, this is how we will help each other and help society. So I'll end there, and thank you.